So I'm Brian Saunders, consultant gastroenterologist at St Mark's Hospital, and this is my research colleague Ahmir Ahmad, and we're very pleased to introduce to you our latest paper entitled Evaluation of a Real-Time Computer-Aided Polyp Detection System, CADe, during screening colonoscopy, the AI DETECT study. Now, we know that the key KPI in determining colonoscopy quality is the adenoma detection rate. And there has been tremendous interest in how AI can help us to detect more polyps, and particularly adenomas. And the latest generation of CAD detection systems are now available. So this study was really looking at an evaluation of this within a defined patient population within the English bowel cancer screening program. So, Ahmir, just tell us the methodology of the study.
Yeah, so this is a prospective randomized controlled trial, which involved all 8 screening-accredited endoscopists in an English National Health Service bowel cancer screening centre. And it ran over a period of around 2 years. It was a parallel design, so participants were randomized to CADe, and we used GI Genius for this study, or standard colonoscopy with a 1-to-1 allocation. GI Genius is the first commercially available polyp detection system, and in practice this involved attaching a module to the existing endoscopy stack, and that fully integrated with the existing setup. And the system highlights polyps that are detected during the colonoscopy in real time and superimposes a green square automatically on the monitor.
And the setting here was experienced colonoscopists?
Yes, all participating endoscopists were experienced.
Within the bowel cancer screening program?
Yes.
All familiar with the CAD system, and in a polyp-enriched group. So the majority of these patients were FIT positive or on surveillance from previous FIT positive colonoscopies.
Yeah, all the inclusion criteria were any patients that met the criteria for bowel cancer screening in England.
Okay, and it was a straight head-to-head randomization between use of CADe or not. Were there any specific things about when the system was turned on, or was it active throughout the procedures?
So as part of the protocol, the CADe system was switched on at the beginning of the procedure and remained on for the entire duration.
So in the CADe arm, endoscopists would have been aware of small polyps during insertion detected by the CADe, and that was included in the intention to treat analysis?
Yes.
Okay. So let's move swiftly on. Fairly straightforward study, really. What did we find?
So in terms of the results, 658 patients were invited. 614 of those were included and randomized to CADe or standard colonoscopy. And of those, 35 cases were excluded from the per protocol analysis where they had poor bowel preparation, where the procedure was incomplete, or where there was any issue with the data. Endocuff vision was frequently used by the endoscopists, and this was evenly distributed between the groups. And a total of 2,104 polyps were identified. On the intention to treat analysis for standard and CADe groups, there was a borderline significant difference in the polyp detection rate. So in the standard group, the polyp detection rate was 79.7%, and in the CADe group, it was 85.7%. But the p-value here was 0.05. And when we looked at adenoma detection rate, there was no significant difference between the groups. However, with a significant polyp detection rate, that is where an adenoma or a cell-ulcerated lesion was detected, that was statistically significantly higher in the CADe group at 79.2% versus 71.6% in the control group. So there was a small but clinically relevant benefit of using CADe for significant polyps.
In this patient group. So what we're seeing really is perhaps a small advantage, but not a significant difference in ADR.
Yes, and in the per protocol analysis, there was no significant difference observed in any of the rates. And in both analyses, there was no significant difference in the procedure time.
So I mean, the early studies looking at CADe have shown quite impressive advantages in terms of ADR, probably 10% increase in ADR with CADe. Why do you think we're not seeing quite this difference? We're seeing some effect, but not the magnitude of the effect from previous that has been described on previous studies.
Yeah, so there's probably a number of factors that could explain why we didn't find stronger evidence of improvement with the polyp detection system. I think the first thing is that the endoscopists within this group had a high baseline polyp detection performance. They were experienced endoscopists, and that would limit the potential improvement that could be gained with a CADe system. So there is a sort of a ceiling effect with little room left for improvement in the intervention group. So I think that's one aspect. And the other thing is that around 70% of procedures were performed with endocuff vision. And so in the majority of procedures, this would have facilitated the visibility and detection of polyps, and therefore, again, minimize any potential advantage from the additional use of AI.
Yeah, so the endocuff vision is fairly standard practice to use within this patient group for detection. And of course, it's complementary to the CADe system, but there's probably, it becomes more difficult to get a marginal increase when you were talking about highly performing endoscopists, but also using the endocuff vision. Was there any downside to using the CADe system? Did we see any difference in duration of procedure or complication rates, or were there any issues?
We didn't observe any significant difference in terms of procedure time, so no adverse effect on procedure time, and no sort of adverse effect in terms of complications from the procedures.
So Ahmir, drilling down on the results, was there any difference in the morphology of the polyps detected in the two groups?
So in patients where polyps were detected, there was no significant difference in the distribution of polyps, the Paris classification, or the polyp size.
So we're not seeing a higher detection of flat lesions, for instance?
Yeah, no, we didn't observe that in this study.
Or serrated lesions specifically?
No.
Okay. So just to sort of give us a conclusion to the study, how would you summarize the results?
So I think what this study has shown is that using a CADe system has a potential benefit, but within an experienced endoscopist group, where the baseline polyp detection rate and adenoma detection rate is already very high, there is a limit to the benefit that an AI system can add to that baseline PDR and ADR. However, there is potential in the future with improvements in the algorithms that there will be sort of a greater role within this group. But for the time being, I think it will be more for less experienced endoscopists or low detectors.
Yes, it's a rapidly evolving science at the moment. There's tremendous interest, and if you look at PubMed, there's a huge increase in the number of papers being produced looking at AI. So it's an exciting time for AI and colonoscopy. There'll be many developments coming through. In particular, we're looking forward to CADe, which may well make an optical diagnosis, resect and discard strategy a reality. And we very much hope that you enjoy reading our paper. Thank you.